All right, so I'm going to be the heavy here because I'm going to be laying down some really nasty looking pictures of what the impacts are and kind of trying to get you all up to speed on Phytophthora at a level that allows you to start thinking about it a little bit more in how we deal with these pathogens in these systems. And there are a couple of things, areas where I may differ a little bit with what Susan had to say. Sometimes it's just a semantic issue. And a lot of people come to Phytophthora from this sort of perspective. They think of Phytophthora as in the landscape, and whether they're really just a problem or over-irrigated landscape plants. Now, Susan's already kind of given you the inclination that that's clearly a myth. But I'm going to show you that not only do you not have to be in a landscape situation, you also don't have to be in a very wet situation to have horrendous impacts. So I'm going to show this picture of Arctostaphylos myrtifolia. This is an endangered manzanita species. This is also one of the last healthy looking plants you're going to see in this presentation. This is what stands of Arctostaphylos myrtifolia look like once Phytophthora cinnamomea has been introduced to them. Pretty much causes mortality over large areas of both myrtifolia and some sympatric Arctostaphylos in that species, in that area, Arctostaphylos glauca. It works its way up hills as well as down. It goes faster down than it goes up. You can see how it advances over time into areas. It just swoops these areas out. And nothing really comes back in terms of these Arctostaphylos. They're pretty well shot. This particular soil type is you almost take a look at the surface, you can see it looks a little bit like desert pavement because it really is a very depauperate landscape. Actually, these soils are very acidic. Just to show a little time progression on this little spot here, one thing that you want to note is that after all this time in these mortality areas, there's nothing's growing back, certainly none of the Arctostaphylos. And in terms of the areas we're talking about covering, this is just south of Ione, all through those entire areas, and that's just a portion of the area that's infested. Pretty much all the manzanitas either dead, dying, or there's just a few remnant islands of it left. So these cover huge areas. So we started working back on that in 2002, and then a couple of years later we got a call. We sort of thought that was really the first wildland demonstration in California that Phytophthora cinnamomea was active in our wildlands. And we got a call from Fish and Game about some Arctostaphylos pallida, another endangered species in the Oakland Hills, much more limited range, where they saw some issues, and we found that both the pallida and giant chinkapin in that area were being killed. A couple of years later, that's just upslope from the Huckleberry Regional Preserve, which is not a good thing because Phytoptheras tend to move downhill pretty readily. We did get a call from a Fish and Wildlife Service biologist a few years back and of another spot, and this has a very limited range, but you can see that there's been infestation in multiple parts of its small range already. And so we started thinking about Arctostaphylos, so maybe they're kind of a special case. Well, we've been working in China Camp State Park since 2000 on sudden oak death, and we had realized we had some other mortality we didn't really pay much attention to because it wasn't sudden oak death, but we eventually realized all these trees that we had sudden oak death taking out the oaks, and we expected these bay trees would just take off because, of course, they had been released from competition, but in fact these trees were dying. And the reason they're dying is because cinnamomea was also in this area. So we have the oak trees being killed by Phytophthora ramorum, and we have bay trees and madrone being killed by P. cinnamomea in the same area. That particular mortality center continues to advance. You can see some recently killed madrones right here. In the park itself, there's a very heavily infested patch here, but subsequently with some other data collection, we found cinnamomea is far up the slope, is up this fire road, And again, everything downslope from these in the watershed potentially at risk. So as I described it to our state parks context, there, it's really kind of a slow motion disaster area because it doesn't occur overnight, but you're going to see major mortality throughout those areas as that pathogen continues to spread. We were working with the PUC on the Peninsula watershed on sudden oak death, and they had mentioned that they had some mortality of madrone in that site as well. We checked that particular area out and found that there was also a Cinnamomum mortality center there. In general, in these coastal forests, we'll see madrone being more sensitive than the bay, but the bay eventually thins and dies out as well. In that particular area, we'll talk a little bit more further on, we actually had multiple species of Phytophthora causing similar symptoms. On this particular spot, both Cinnamomum and Cambivora were present. A little closer by, the Mount Tam watershed. We've, again, areas where we were looking at sudden oak death, but we've noticed some mortality of madrone. This is a canopy of this large madrone which has been killed. These are P. Cinnamomum infected areas as well. You can see that these trees go out takes a couple of years, but they'll be thinning and then eventually are killed. And again, we're not talking about really wet sites. These are up on slopes in this situation. Here's another patch further down the hill, and you can get an idea that these are not really lush foresty areas. They're a little bit depauperate soils, and it's really just devastating in these areas. We were interested a little bit also in chinkapin. We heard some reports on mortality in chinkapin, and we had noticed in the open hills that we had dying chinkapins associated with mortality with Cinnamomum, and sure enough, this is another spot in the on the Mount Tam watershed, not on MMWD land, but we found a large patch of chinkapin there being killed by Cinnamomum as well. One of our more recent wildland finds was in Jack London State Park, another site we've been working at for a long time, and it turns out that there's a mixed mortality center there, which includes both Cinnamomum and Cambivora, again attacking mostly the bays and madrones, but adding to the grief there, which is also another area where P. remorum, by the sudden oak death pathogen, is found. And here's just another example. This is a kind of wildland site in Sonoma County. Here, it turned out there was these little patches of dying manzanitas, and somebody had been aware of our other work and had called us. Well, we were able to confirm P. cambivora associated with a number of these mortality centers. The interesting thing is most of those were in areas where they had dug test pits for their percolation tests. So of course the equipment was going around from one spot to another, and probably that equipment was originally contaminated from where it came from. And just about every place where they'd been working, we had these mortality centers. Back in the Peninsula watershed, just to spread it around, as we mentioned, as Susan mentioned, there's a lot of other species of Phytophthora that don't actually have names yet because they keep finding new ones. This one's just called taxon PG chlamydo. But we found here mortality of coffeeberry and coast live oak. Not a good combination. Again, this happens to be a slightly wetter area than a lot of other sites, but it's just kind of problematic to see that we've got an awful lot of types of plants that can be killed in good patches. To throw in a few conifers, this is also in the watershed. This is a plantation of Douglas fir, which has been declining and dying out for a number of years. And we found Cryptogaea was associated with a number of these dying trees, and Cryptogaea is a well-known pathogen in forest nurseries, so it's not really so surprising in a way that it might have gotten moved out here. So what we wanted to point out to here is that we're not talking about just wet areas, we're not talking about just inland areas or coastal areas. Bottom line is we've gone a lot of different places and found mortality centers. So we know that it's out there in a lot of places and causing damage. So it's not theoretical in California, this is happening right now. So is this a myth or a fact? Are they everywhere? And this is where I'm going to say that really is a myth, because in general, the most destructive Phytophthora you have, the ones that are causing mortality, are all introduced species. They're not something that was here, and that kind of gets to the aboriginal thing. Pre-settlement, these species were not here. So it didn't matter what their burning regime was in that respect, because if you don't have the pathogen, you don't have the disease. But once you get them in and start spreading them around, then you actually find out where the susceptible vegetation, the susceptible types of environments are. Just to give you an example, this is an Ione manzanita transect, one we established a number of years ago, where we're actually sampling along this transect to recover the pathogen. You can see pretty much a dead line there. Well, if you take from the dead line out, you can recover the pathogen. Go from the dead line in, as little as 2.5 meters, you don't really recover the pathogen. Basically, what's dead is where the pathogen is, because this is such a sensitive species. Just for comparison, this is a 2014 picture. This is that same transect. The disease has moved uphill. It's well past the 5-meter mark there now. So you can see it just on its own will continue to spread from root to root contact and spread in the soil. But it wasn't there when the original picture was taken. So we're looking at spread rather than a situation where it's everywhere and suddenly the conditions change. So where are these Phytophthoras coming from? We've already alluded to nurseries, nursery stock, and here's a site where there's probably a pretty clear connection between some landscaping that's been done here and the mortality of these bay and madrones in this area. Again, both of these are cinnamomum related mortality, adding to the woes of this particular property, which also has a lot of sudden oak death. Back to the peninsula watershed, one of the things we did when we tried to survey out and delineate this mortality center with the madrones is we actually found there were quite a few different Phytophthora species in little points along the watershed, a lot of them along roads, and there was a lot of clustering around the Filoli estate, which is a historical estate. In those two areas, there's a lot of traffic between those two spots. As you know, they have a nursery, they have all these ornamental plantings, they actually know that they have Phytophthora diseases in those plantings. And you can see that where there's more traffic back and forth between this area, we get more density of a different variety of species. But even note that these white circles are negative isolations, areas where we might have seen some kind of plants that look potentially symptomatic, and these are negative. Again, you don't have to get far out beyond a mortality center to actually come up with a spot where they're just plain not infested. So the idea that Phytophthoras are everywhere and you can't do anything about it is something I'd like to lay to rest early in this presentation. And in fact, these agencies and others have taken this seriously, because here's a situation where you have a roadside and you can see that it's mowed. Whoops, I'll back up in a second. It's mowed along that area. That occurs during the wet season. Mud from this infested area can be moved further up and down the road, spreading that mortality center. So PUC is taking steps to change the surface on that road, control traffic in there, try to avoid spread in that area. Other agencies also taking this seriously. At China Camp, they completely closed and obliterated one of the fire trails that went up the slope through the mortality center because of P. cinnamomum. The BLM has a preserve with the Ione manzanita on it that they've closed to public use, partly because they want to try to avoid further spread of the disease. So these are things that are impacting areas in California right now. So let's get back up and get a little thing about biology here. This would be a good song title if we were making Phytophthora songs. But so what is this thing you can call Phytophthora? Well, it's not a fungus, and that's one first thing you have to kind of wrap your head around. It's really a fungus-like organism in the group called the water molds. These water molds are actually more closely related to algae and diatoms. They're in the group that gets various names in the big family tree, the Chromista or the Stromenopiles. There's various names there, but the bottom line, they're not really very closely related to fungi, even though they have a similar growth form. There's some big differences. primarily their mycelium, or this white fluffy stuff on the plate you see, is diploid as opposed to the haploid state of most fungal mycelium. It doesn't have cross walls. The cell walls themselves are made out of beta-glucans or cellulose, not chitin, which you find in the true fungi. So the cell walls are quite a bit different. And most of these, or most Phytophthora are pathogens, and they're pathogens of dicot primarily. We've already talked about some. One thing about P. ramorum, which I want you to kind of get your head around, is P. ramorum is one of the aerial Phytophthora. Not all Phytophthora are aerial. It moves because its sporangia can break off and get splashed and works largely above ground. Most of the pathogens I'm going to focus on, like Cinnamomi or Cactorum, these are soil-borne pathogens, and mostly they're confined to the soil. In nursery situations, sometimes you can get them splashed up onto the leaves and stuff, but in general, we're talking about soil pathogens here. As you can see, these are all very destructive, and in fact, the name Phytophthora, it comes from the two Greek roots for plant and destroyer. So these are the plant destroyers. And again, although these are very locally famous, probably the most famous, as alluded to, is potato late blight. You will not see memorials that are related to other plant pathogens, but this one you will, because of the great impacts that P. infestans had on Ireland during the late 1845 to 1850 period. Okay, Susan kind of talked a little bit about numbers of Phytophthora species. There's some databases that people are just keeping sequences piled in just to try to keep track of things. Clyde Brazier, one of the world authorities on Phytophthora, produced this graph at a conference in 2007, and showed how we have this number of increasing number of taxa that have been defined over time with this kind of steep, sharp increase at the end. He speculated there could be up to 200 to 600 species out there that we just don't know all of them. But if we add this extra taxon in the Phytophthora database and extend that line out, we actually see it follows pretty much on what Clive's line was showing. As we learn more and as more of these things get out of their native habitats into other places, we're just seeing that a lot more of them. Okay, some basic biology. So we've been talking about mycelium, and we talked about one strand of mycelium as a hypha, but this is what we see that's growing on media if we isolate it, or growing through plants if they're infected. They can take various forms, sometimes more gnarly than others. Sometimes mycelial characters help you identify the species. Asexual reproduction occurs primarily by means of sporangia, and these sporangia are produced under moist conditions. They can germinate in two different ways. They can have what's called direct germination, where they simply put out a germ tube and produce more mycelium, and that potentially infects something. However, the way that they really get around is by means of zoospores, and we have a cheap animation here. Zoospores are swimming spores, and just because I don't have a quick way to do it, you can sort of see that's what's happening. I do also have these zoospores actually have two flagella. That's how they're swimming around in the water. They are released only under saturated conditions, so they're not going to, unless you've got free water, they're not going to be released. And if this movie runs in the next one, this just shows you a little bit more of what's happening in real time. So these sporangia, sorry about the low res because of this PowerPoint didn't like to deal with higher things. So basically, they pop out of these sporangia and they go swimming around. There can be up to a couple dozen in some of these. There's going to be another one here that's going to blow in a second. There goes all those sporangia, those zoospores swimming out of that. Okay, this is just one little teeny piece of root, right? There's probably maybe a dozen different sporangia in view here. There could be thousands on any given root that's infected. And so each of those releasing a couple of dozen swimming zoospores. That's how we get rapid buildup. What happens to these zoospores? Well, they essentially are chemotactically home in on roots where they can encyst sometimes in great masses. Once they encyst, they form these germ tubes and they penetrate into the host. So as we see here, there's a zoospore cyst on the surface, it's sending its hyphae down and it's starting to kill the host cells below it. So that's how they get around it, ramifies in through the host and eventually produces various types of spores, including sporangia again. It can also produce things like chlamydospores, which are survival and can function for infection as well. Or another type of survival sexual spore, the oospore, which is where the group, the oomycetes, gets their name. So we can, if we break this down into kind of a disease cycle or a life cycle, this is the fast operating portion of the cycle where you have, under wet conditions, you can have rapid disease cycling. You have infected roots, sporangia produced on the surface. When free water is there, these zoospores are released, they swim around, infect more roots. This whole cycle can occur in 24 hours. So if you start with a very small infection, you can ramp it up to a lot of infections and a lot bigger infections in a relatively short period of time. Okay, now these are great for dissemination and dispersal under wet conditions, but if it's dry, the pathogen also has to survive. So that's where we kick into the oospores, the chlamydospores, other kinds of survival structures. And under dry conditions, in root fragments or infested soil, you're moving around oospores or chlamydospores, and not zoospores. But these can germinate in some cases either directly, like chlamydospores can directly infect roots, or they'll form sporangia, which then are released under wet conditions to start this whole part of the cycle going. Now we take advantage of this release of zoospores in one of our kind of common ways to detect Phytophthora in soil root samples, and that's baiting things. So we can use a couple different types of baits. Here you're seeing some leaf disc and a green pear. We can also use complete leaves as well. What happens is you float those out in the water. The zoospores are being released, and they infect these things. So you end up with lesions that you can then isolate from. So here's an infected rhododendron leaf, and around the edge of this pear you can see all these brown infections. It turns out green pears are really nice because if you've got a really hard, solid green pear, there's not a lot of other things that will infect it. Phytophthoras are almost about the only thing, so you get a pretty selective thing. And you can also get a kind of semi-quantitative kind of dose response thing. If you've got a lot of infection on your pear, you've got a lot of inoculants that's right there, but you bring it down to sometimes just one or two spots, but at least it shows you you've got presence in that site. Okay, so we know this is a water-borne pathogen in situations, and so sometimes this statement is made for simplification. That really is a myth. Okay, Phytophthora doesn't come from overwatering. So this is the bottom line. If you don't have the pathogen, it doesn't matter how much you water it, you're not going to have the disease. So this is the thing we want to, and this statement was made recently by a horticulture professor in some email exchange. I had to jump all over her for it. And she said, well, I didn't really mean it. Of course you didn't mean it, but if you're talking to people who might not understand that you don't mean it, that's a very dangerous statement to make. And here's just a quick illustration of that. This is actually in a pathogenicity test on my own manzanita. We had 2 rows of plants here. One row is being put in these double cups because we flooded them for periods. And you see this set is all dead, the other set are all alive. Well, that's because these are the non-inoculated controls on the top. Those are the inoculated ones below. Flooding wasn't enough to kill these things, but with the pathogen there, certainly that provides opportunity for infection. Okay, this brings me to the famous plant disease triangle, which is that we really have several factors that come into play when we have disease, and the classic 3 are the pathogen, a virulent pathogen, a susceptible host, and a favorable environment. And basically take one of those things out, make them unfavorable, and remove the pathogen, you don't have disease. So that's straightforward enough. We can complicate this a little bit by really realizing we have both an abiotic environment and a biotic environment, things like mycorrhizae and antagonistic bacteria, et cetera. And there are interactions between all of these things. They can influence each other in various ways. So really what happens is optimal disease occurs when you kind of hit some sort of sweet spot where everything is really favoring the pathogen over the host, and that's when things can really ramp up. But there's still the element of time in that. You have to have, the more time you have under those favorable conditions, the more likely you're going to get disease development. So let's break this down just for Phytophthora. Okay, what kind of abiotic environmental features are really beneficial? Well, humidity and moisture are there for, especially for the explosive fast generating part of the cycle. They really don't like, these are very aerobic organisms. They don't really like saturated conditions for long periods. They really don't, they're not at their best in stagnant areas. They're really best with aerated soils. But they do like periodic saturation and relatively moderate temperatures. Well, I wonder what kind of situations would provide that. Well, you've got all those things in a typical nursery situation. Well, what about the host factors? Well, high host density, okay, check that off. How about high root density? Well, we got that. You never will see roots as dense as you find them inside of a container. That just allows cycling to occur a lot faster and more infectable material goes by. And predisposing stresses do occur in nurseries. You can, during your watering cycle, you can expose things to periodic drought or excessive salt, heat. All those kind of factors can reduce the resistance of these hosts. In terms of the biotic environment, which if it's really intense, can inhibit disease. Basically, if you have a lower density and diversity of these antagonistic microorganisms, you're less likely to have disease go. Well, if we think about how we use artificial media and very simplified systems, of course, we're not going to have this complex, very complex microbiota in there. So that's the type of thing that is going to be favored. It's not really going to prevent Phytophthora from running in these things. Finally, the element of time. Well, the longer your plants stay in the nursery, the greater you have for time for things to infect repeatedly. So you add this all up, it really comes down to what I think Susan was trying to get to. Nurseries are almost, if you're trying to design an ideal environment for Phytophthora diseases, you would end up with a nursery. Okay, so that means that really the basis of everything has to be taking the pathogen out of the nursery, because if there's no pathogen, you can have all these features and that's fine. Without a pathogen, no disease. You add this into that environment, boom, we have a real problem. So inspect your roots. We like to say that to say, okay, you have disease, you just look at the roots. Well, you can't always tell, especially when we start working with native plants. They have a lot of different looks to them. So here's one of these Ione manzanita. You can see the stem of this inoculated plant is all necrotic. So all those roots are all brown and they don't look very good. Compared to this healthy plant here, where the roots, well, they all look brown and they don't look all that good either. But if you looked at it closely enough, you say, well, the density is less here and there's more broken off. The differences are more subtle than you would expect. Here's another plant. Okay, is this the diseased one or is this the healthy one? What do you think? There's a white roots down there. Looks kind of brown up there. What about this one? Okay, that one looks healthy, right? But if everything in your nursery looked like that, they'd all look healthy, wouldn't they? So that's one of the issues. If you don't have clean stock to compare with, it's pretty hard to tell. And even in infested material, there's some nice looking roots. I mean, there's some white roots in there, white colored roots. So to think you're going to inspect your way out of this is a little bit not quite there. And this has been a kind of a thought, well, native plant, horticultural nurseries, they're the problem. It's not these native plant nurseries. Well, as it turns out, unfortunately, that's not the case because with a lot of work done by ourselves and others, We know that there's a lot of Phytophthora out there in these nurseries and has been for years. This was work done a number of years ago. We were doing the pathogenicity test for P. cinnamomea madrone. Elizabeth Fitchner in the Rizzo lab had to get some plants to do these tests on. Well, she baited these plants before she used them. And she got 8 Phytophthora species from a very small batch of plants from three different nurseries, okay? And a lot of these are, one of these is the pathogen we were doing the pathogenicity test on. And we looked at this and we thought, well, of course, other nurseries are infested. Why wouldn't these native plant nurseries be infested? And that turns out to be the case, unfortunately. So when we were working with PUC on some of their sudden oak death related stuff, they had indicated they had a big restoration project going on. And we suggested, well, this would be a good time to make sure that besides not moving worm around, you're not moving these other soil pathogens around. And we got a plant that had just recently been planted at one of these sites at the beginning of this year, which turned out to have been infected with not just P. cactora, but P. tentaculata, which Suzanne will be talking about later, which is basically a quarantine Phytophthora, not just run-of-the-mill stuff. This is something that has only been detected in California once before this. So that was not a good sign. Here's one of the unplanted nursery seedlings from the same thing. It also had cactorum, more cactorum than tentaculata in these. This thing hasn't had functional roots for probably a couple of months, right? It's so rotten. But it's not even totally dead yet. You can see it just collapsed recently in some January heat. But this is one of the issues in native plants, is a lot of them are very drought tolerant. So if you have very drought tolerant plants, even if most of their roots are non-functional, they don't die very quickly. And so you might think, oh, okay, these plants look good, and out they go, and then next year they die. Well, maybe we didn't water them, whatever. This causes some issues. So we did do some additional inspections. This particular nursery, we picked up P. quercitorum on oaks. That's another species that has hardly ever been reported in California. We looked at this nursery, which had some carex. Well, carex aren't really considered to be Phytophthora hosts, but we said, okay, well, we're here anyway. We'll sample, we'll take some of these ones that didn't root very well and do some isolations there. We got Phytophthora plurivera out of there. So we're thinking, well, is it the soil just so heavily infested, or is it actually a host? This doesn't even belong in there, really. But then you can cling to the idea, well, they won't survive when they're planted out in the field. Hate to burst anybody's bubble on that one, but the fact is they do survive when they get planted out in the field. And we have plenty of evidence on both our work as plenty of other work, and Susan a lot alluded to that. But here were some toyons planted at one of the sites with P. cactorum on them. They were still in these bags because the roots never really grew outside of these bags because they were so rotten. Here was some older Mimulus or Diplacus. We actually had 2 different Phytophthora species at that particular site. This was a problematic site because just before we got there to sample it, there had been a big shower and there was a lot of standing water and it was wet. And that's really not a spot where you like to see Phytophthora. We did find several species of Phytophthora on some plants that we didn't really think were very likely hosts either. And we also picked it up in the water that was over this road, which was downstream from where they had their holding area, where they had been holding their plant material. We had previously sampled further up this watershed and not found any Phytophthora. Here's another large planting. You can see a large density of material in this particular site. Mimulus that had been ground 2 years had P. tentaculata, as well as there was other 2-year material. So we know that it overwintered and oversummered in these sites as well. So it didn't go away after it was planted. Some sites in San Jose. This is a big re-veg project in here. We're looking for things on Mimulus, tentaculata in particular. We walked past this nice coffee barrier that was recently, it's been in the ground for obviously a while, but it was died recently from cactorum. and we came across this mimulus with this Niederhauser eye on it, which is yet another problematic species. So you start getting a concept here that there's a lot of stuff working its way out there, and this was kind of one of those add insult to injury type of things. Here's this nice Ceanothus endangered species. It's being planted out on an assisted migration project. Well, this material turns out to be pretty heavily, almost uniformly infested with Phytophthora, and most of it is dying. So we have the potential of having infested this whole outplanting site. We've mentioned pair baiting. This is some of the pair baiting from that particular. So you can get an idea. We're talking about pretty heavily infested material. So just in some work that we've done, and we haven't really been at that systematically or carefully, but basically we've got 10 species of Phytophthora here on all these various hosts. These come from 6 different nurseries. You can see they're on a wide variety of plant species. These blue ones are monocots, things we wouldn't even consider to be hosts normally. We've got detections in the nursery, we've got detections in the field. Not a good scene. Our box score for the nurseries will remain nameless, but are identified by arbitrary letters. We can see that we had multiple species in most of these things where we took more than one plant, for example, and the red species are things that are unusual in California. They're just not out there, and so that means they're showing up pretty commonly already in some of these nurseries. Well, okay, so let's get into nursery practices. My plants are off the ground, so I'm not going to have a Phytophthora problem, right? Well, it's a good start, but that may not be everything as we can think. So here's a site where they did take the material off the ground. It was on this material before, but if they just put it up on to something else and it was already contaminated, it doesn't really make it go away. So that's one of the issues here. The other issue is that if you're only using something like flats here, A, they have flat surfaces there, so there's still connection of water between material. And B, typical splash dispersal just from raindrops and things like water drops is about at 1 to 3 meters horizontally and ½ meter vertically. So certainly you have potential splash right off the ground onto these materials. And once anything gets infested in this block, it's going to go to town because of lateral splash. Furthermore, even if you start out with them higher up, if you start out with contaminated soil in some way, then it doesn't matter how high you put it up. You can put it up over your head, and it's still not going to stop the fact that you've got Phytophthora already in your soil. So maybe I can test my media and tell whether it's free of pathogen. Well, this is our first one where we're going to say it's not so simple. We can detect Phytophthora in soil, but only if it's at pretty high levels, because this is a sampling issue, it's statistics. And then we also can have media infected at different times. So we test it at one point, it gets infested later on. That first test is meaningless, because now we have an infestation further down the pike. And all these tests can provide false negatives just from not hitting the right spot of soil. So here's a little nursery that was doing its best getting its stuff off the ground and trying to implement some BMPs, but how do they get the soil out of this bin? They got to walk in there, right? So even if that material was autoclaved and put in there, how long is it going to stay potentially clean? Maybe not so long. How would we really detect real low levels of inoculum if we were in a large volume of soil? This is a difficult statistical problem. Well, if I was setting up a study to do it, I would do this. I would take a lot of that material, put it into containers, and I'd plant susceptible material into those containers. So all I would have to do is then look to see where I might have the odd declining plants. Well, isn't that kind of what we do in nurseries in general, right? So really, nurseries, besides being very favorable from all those other reasons, really have the statistics working them against them. Even a low level of infestation in the process could blow up very quickly because you've got large batches of material. So here's a myth. Well, maybe we can just apply fungicides or biocontrol agents and get rid of them. These are suppressive materials right across the board. And even for things like biocontrol agents that might actually directly kill mycelium, in practice you never get 100% kill. So at most you're suppressing things, but you're never eliminating the pathogen. So here's a sensitivity test we ran to check to see whether some particular pathogen was susceptible to metalaxyl, a common and very effective fungicide. And what you see here is that just growth on agar, is eventually almost stopped, but you have to go up to some pretty high concentrations to do that. How those compare to label rates? Well, the residue tolerance range for agricultural crops is in about that range, up to about a little bit over 1 microgram per mil. So pretty much when your field rates are going to maybe approach this level, residues in the plant. So clearly you're just suppressing the material, so it may not sporulate very well, but it's there, and once the residues drop, the pathogen can come roaring back. Now, one of the issues with another issue using these materials in nurseries is you can have some pretty shabby looking material that you can't isolate material, you can't get pathogen out. So you kind of game the system a little bit, if you want to look at it that way, by putting on these fungicides, because now we go in there and test it, like we got negatives on these things, but clearly they're really rotten plants. And the bottom line is you're actually better off not using these fungicides and detecting things earlier rather than suppressing them and having them spread around insidiously. Okay, well maybe we just have garden variety pathogens. Maybe we aren't really too concerned if they get planted out. Well, I would say that that's not really the case, because most of our really heavily destructive things are really not regulated by USDA or CDFA. Those regulations really have to do with commerce and agriculture and other things. And so they're really not going to protect you against these pathogens in terms of regulations. And I will put to you that restoration really doesn't involve in introducing exotic pathogens. If we just use our sort of Wikipedia definition of restoration, and this is a little restoration project all planted with acorns, so no chance, and non-irrigated, no chance of introducing pathogens. I think that fits the kind of definition you're looking for. What happens if you're introducing these pathogens in your restoration project? Well, not only is your project not going to do very well, but then you're having the chance of expanding it into the rest of the environment as well. So I'm going to really suggest that this is the guideline for restoration. If you're really restoring things, you can't be putting pathogens out there that can destroy the ecosystem. So that's a really zero tolerance type of situation. So to kind of summarize, why should we be concerned about these pathogens and habitats when we're doing them? Well, contaminated nursery stock, obviously a very efficient way to move pathogens out into the environment. I mean, just if even a small fraction of this material is infested, we're spreading it all over the place and giving it lots of opportunity to go. There's an increasingly wide variety of Phytophthora species in these nurseries, both native plant nurseries and horticultural nurseries. Jennifer Park in Oregon recently completed a year-round intense study on 4 nurseries, where they had 28 taxa of Phytophthora in these 4 nurseries. Okay, it's just insane, really, if you think about it. And that material is all going out and being planted places. And so if we look more intensively, we can find more, just because if you don't look, you don't see anything, right? That's pretty straightforward. So it's a problem there. We already know that these introduced species can have devastating impacts in our native habitats, in a wide variety of habitats in California. They're already doing that. And we have both rare species like these manzanitas, or common species like bay and madrone, or Doug fir, if you want to look at it that way, it can be affected. So we have a lot of species at risk. When we get these things into the landscape, it's a lot easier for them to get moved around by means of soil movement. We talked about those oospores and chlamydospores in the soil. You start moving around either on boots or on larger operations, you can move a lot of contaminated soil, mess up a lot of areas really quick. As I mentioned, because we have these motile zoospores, and under wet conditions, if you're having zoospore release, They can run right down slope in surface flows. They can end up in creeks. They can move a long way down slope very quickly. So if we're doing restoration projects that are in upslope areas, we have put a lot of potential areas at risk downslope that might have other existing vegetation. We also have this problem that these are very persistent in soils. This is some of my own manzanita, and you see some old dead plants here. These brown things are actually seedlings that came up afterwards, and they lived for a short while before they encountered some other old inoculum in the soil, and they were subsequently killed. That's why we're not seeing manzanita come back in these areas, because these spores are long-lived in the soil. You get other hosts that, including the same host, if it regenerates, it gets killed again, more inoculum, more survival. That's really very devastating. And as we get into bigger and bigger areas that are infested, like these are the infested areas in Jack London State Park, you're not really going to be able to eradicate in those areas. The best you can do is now start to manage and try to limit further spread. So we have a whole raft of issues here that we want to have to deal with, and we don't really want to deal with these situations in our wildland. And so this is the last thing I'm going to pose in terms of this myth or fact thing. Is it really too late? I don't think so. And the fact is, we still have very limited areas that are affected. But if we want to do something, we've got to start doing it pronto, because these areas are only getting more and more infested over time. As Susan indicated, Phytophthora have huge impacts in many other parts of the world, and we don't really want to get to those situations. This is just a portion of Western Australia where Phytophthora cinnamomea is causing dieback in a wide variety of species. And we're not there yet, and we don't really want to get there. So that's why we're here using that little catchphrase. We're just here to spread the word and not spread Phytophthora, hopefully. So areas that have been impacted, we may not be able to do much with. This is part of this is actually a fish and game preserve, especially for these endangered species. Well, all this habitat area is pretty much destroyed here and not going to come back. But there's still some island manzanita along the edges here. Can we protect that? Can we keep it from spreading? We've got some studies in place on that particular issue, but that's, as we eat up more and more habitat, especially for endangered species, eventually you run out. And when you run out, you don't have any options. So this is not a new problem, okay? We've known for decades that these are issues, and I think really the issue is a lack of awareness rather than a lack of information. We're going to talk about BMPs later in the afternoon, just to show you that BMPs have been around for a while. These over on the right are a little bit older than I am. And this is spoiler alert, but when it comes down to is this, you need to start with clean material all through, and you should keep it clean. If you do those two things, you end up with clean material at the end. So in some ways, it's not rocket science, it's not impossible, but it requires eternal vigilance. It requires a high level of thought engagement by everybody in the process. So I'd just like to thank kind of the wide variety of agencies and people that have been involved in different parts of this research that we've been doing. And I think I have a little bit of time, so I can probably take a couple of questions. Kathy.
Do oospores and chlamydospores live in the soil in the absence of any plant tissue, like just in dirt?
In free soil?
Yeah.
Okay, the question was whether oospores and chlamydospores can survive in the soil in the absence of clinging plant material. I think the answer is yes. That's kind of a hard thing to study, partly because they're bloody tiny, and the only way you can get them out is by doing all kinds of sieving and everything else, so you don't really know whether they were totally free in the soil or whether they were still clinging to some of the plant material. From a theoretical standpoint and from what, you can certainly add chlamydospores and oospores to soil to infest it too. So I think the evidence would be that, yeah, they can survive. But generally, they're often going to be associated with some dried up plant root material that they're either going to be embedded in or maybe on the surface of.
Are there other materials besides container stock that we should be concerned about bringing into sites like compost and mulch?
Okay, good question. Are there other materials you should be concerned about bringing to your site besides container stock? Well, container stock are kind of they're the chief typhoid mare because they're, as we've gone through, have the highest risk of being contaminated. But what about other materials? Well, okay, what about sand? Well, sand can be contaminated with Phytophthora, depending on its source, where it's mined. A lot of river sand downstream from agricultural operations, it can be detected in those areas. Mulch, a lot of mulch is forest product material. Okay, where's forest product coming? Well, if it comes from a forest area that's got an infestation of earthworm or other pathogens, cactorum is common in a lot of forest areas, you may be bringing in on that as well. And commercial, well, say recycled green waste mulch. If it's been composted to California state composting standards, generally it should be free of pathogens as long as it hasn't been recontaminated in the process of shipping it off the pads and moving it elsewhere as well. So you've got all these things. So yeah, you have to really pay attention to all these things if you're going to try to keep from moving these pathogens around. But if you don't start with the nursery stock, which is like flaming red and burning, and put that out compared to these match heads, then you're gonna have a big problem to start with. I mean, you can start a fire with either a big brand or a little match, but it's going to be a lot easier if you've got the big torch. Okay, so the question is whether as we find more hosts, do we see less virulence? And probably the best example of something along that line is Phytophthora ramorum. Phytophthora ramorum being an aerial pathogen and very lethal, exerts a lot of selective pressure against susceptible genotypes. And so you do see that the first things that are killed are the most sensitive genotypes out there, and there are some genotypes that will stick around even under some amount of attack. So you will have, and hopefully that's what happens in things like tan oak. That's really our only hope, is that there's enough resistance in the population we eventually select for a resistant population. But you may not have the genetic width in that species. We look at island manzanita and the other manzanitas with it, it's just blasted everywhere. There's no survivors. And the stuff that comes back up is killed. So there's not enough genetic base there, or they're just too innately susceptible. And they weren't co-evolved. Remember, the Cinnamomea is probably native to either Southeast Asia or Oceania. It's not from around here. And these things during their entire evolutionary history have never had to deal with this, so they don't have the defenses out there. Question?
What's the regulatory framework in California for commercial nurseries? Is there something that helps in that respect?
Well, okay, so I should let somebody else say it, but the bottom line is there's not much. What it comes down to, as Cheryl I think has said before, you can have a little of a little, but you can't have a lot of a lot. Okay, so the nursery cleanliness standards, essentially, unless you're dealing with a quarantine organism, basically, if you have a little Phytophthora in there, they think, well, okay, well, that's how nurseries are. But that mindset is based off of a whole set of thought that the client here is the nursery. The nursery industry is the agricultural client. So we're trying to allow them to do their business. We don't want them to ship off a lot of very contaminated material, but it's too hard for them to clean up their act altogether. So if they have a little contamination, we're not too worried about it. Okay, maybe that works for landscapes or something like that. For restoration, it doesn't work at all, because you say, oh, well, you can have a little Cinnamomea in your stuff, you can plant it out there. Bang, you've just infested a water shed. So that's where the regulatory stuff isn't there to stop it. It really has to come from demand from the consumers of this material. The agencies that use this basically have to specify we got a zero tolerance here. You have to document this. And the nurseries, that's what is requested, they can produce it. They just have to go there. And it hasn't happened yet. And we're hoping through this meeting, and other efforts coming from this, that's where we're going on this. It's just, if not entirely voluntary, at least a structured approach to make sure that we're planning out a nursery stock and wildland situation is not going to have Phytophthora in it. The question?
Since we have this like mapped infestation sites and public lands that are used for recreation, is there like a ramping up of effort to do public education about like ways that things are transmitted?
Well, yes and no. The question is there public education going out on these wildland infestation areas? And it really is kind of spot by spot. At Jack London, we basically just found out this year about it, and now they're thinking, okay, well, maybe we can do some things about trail access and whatnot. Basically, most agencies don't want to deal with it. They hardly got any budget to begin with. To wrap their heads around another whole level is very difficult. But because there's been sudden oak death and some agencies have kind of gotten into the idea of quarantine and trail use restrictions and things like that, there is some hope that some of that is translating out there. But not everybody is doing it yet. So you could, where that chinkapin dieback spot, that was right on a trail. I could walk right through it, right through the center of it, and track Cinnamomum both directions quite easily. And I have the stop sign up, so we'll catch other questions later.